Welcome everyone to this session on trainee research and innovation. My name is Alison Brown. I'm the Director of Strategy and Translation for Medicine by Design. And I'm just delighted to be a part of today's truly landmark anniversary. I'll start by just telling you a little bit about myself. I've been part of the University of Toronto community for over 20 years, starting as an undergraduate student and then going on to study my PhD here. And I've spent the last 15 years or so working at the intersection of research and commercialization. So I'm just thrilled to be a part of this panel. And with me today, I have three of Medicine by Design's brilliant graduate students, Emily McGaw, Sean Kinney, and Sarah Colpitts. They're going to be talking to you today about their avenues of research. They're all working in the field of regenerative medicine and are working towards a common purpose, and that is to lay the groundwork for future transformative treatments for diabetes. Before we jump into introducing you to our panelists today, I thought I would just take a few minutes to tell the audience about Medicine by Design. And actually, Shayna Kelly at the previous panel set this up nicely for me. So thank you, Shayna, if you're on the line. So Medicine by Design was founded in 2015 by a transformative investment from the federal government to launch Medicine by Design, building upon decades of excellence in this space. Stem cells were actually discovered at the University of Toronto by Ernst Till and James McCullough back in the 1960s, another great discovery for the University of Toronto. And at Medicine by Design, we are harnessing the regenerative capacity of the human body to target chronic, devastating diseases with the goal of either preventing further degeneration or, in some cases, providing curative solutions for diseases. The community consists of over 140 investigators, and notably about 900 trainees, so graduate students and postdoctoral fellows, have been involved in Medicine by Design's team projects. All of our team projects really operate with the underlying theme or principle of convergence. So we have on each of our teams biomedical engineers, clinical medicine, scientists from various backgrounds working on transformative regenerative medicine projects. Our goal moving forward is to continually strengthen the Toronto and Canadian ecosystems to ensure that we can continue to lead in the regenerative medicine space. And so Emily and Sean and Sarah today will be talking to you about some of the research that they're working on, namely using cells as living therapies, which is one aspect of the Medicine by Design research portfolio. We also have a number of projects that look to develop platform technologies that can advance regenerative medicine therapies. Examples include solutions for manufacturing and scale-up of cell-based therapeutics. We also have a number of teams that are looking at how to understand and eventually disable underlying triggers of disease, such as inflammation, and also teams that are looking at approaches to harness the body's own capacity for repair through a number of different small molecule therapeutic approaches. And as you can appreciate from this image, we're working on a number of organ systems, including the pancreas and therefore diabetes. Just to end off this brief introduction, I thought I would highlight that in addition to the University of Toronto and our affiliate hospitals, we work with a number of leading partners, both industry partners and not-for-profit partners in the commercialization space. Blue Rock Therapeutics, which was recognized yesterday evening in Gordon Keller's award at the Bloom Burton Awards Gala for his co-founding of that cell-based therapy company. And that's a great example of the type of impact that the deep investment in regenerative medicine is having here in Toronto. So before I go to our trainees for their introductions, I just thought I would leave this brief quote up for a moment from Gordon Keller. When I started this work, I never thought that you could make 1 billion heart cells in a dish and treat somebody with them, but it's not that far away now. And I'd like to hope that as you hear and appreciate the stories that Sarah and Sean will tell over the next hour, you'll also see that perhaps a treatment for diabetes using a regenerative medicine strategy is not that far away now. So I'm going to stop sharing my screen and kick things off with a round of introductions with our panelists. So Emily, who's a developmental biologist, will kick things off with you. And then we'll go to Sean, who's a chemical engineer, and then to Sarah, who's an immunologist. Emily?
Hi, so my name is Emily, and I'm in my 6th year of my PhD at the University of Toronto. I do research in Dr. Maria Christina Nostro's lab at the McEwen Stem Cell Institute.
Hi, I'm Sean Kinney. I'm in my 4th year of my PhD program in the Department of Chemical Engineering and Applied Chemistry in the collaborative program at the Institute of Biomedical Engineering. And I'm in Professor Michael Sufton's lab for tissue engineering and regenerative medicine.
Hi, I'm Sarah. I'm at University of Toronto in the Department of Immunology, and I'm in my 2nd year of my PhD in the lab of Dr. Sarah Crome under the Toronto General Research Institute.
Great, so I'm going to start with Emily asking a few more questions about what gets you excited about the pursuit of science and research. I mean, really, how did you fall into doing a PhD in regenerative medicine here at the University of Toronto?
So I was actually first interested in doing diabetes research when I was in my 1st year of undergrad at university. My sister was diagnosed with type 1 diabetes. Even though I was studying biology at the time, I actually didn't know a lot about type 1 diabetes, nor did I really understand what it meant to live with type 1 diabetes. It didn't take me quite long to realize the challenges that are associated with this disease. And although my sister is great at managing her diabetes, I know it is quite tough. So at that time in my education, I thought I'd give research a try. So I actually first started off as a summer student. So I approached Dr. Nostro, who does diabetes research, and I asked if I could do a summer position in her lab, and she agreed. And at that moment, I fell in love with research and I loved it. So I actually went back to her lab as a master's student, and then I did my PhD in her lab. So I've been in her lab for over 6 years now doing diabetes research, and I've loved every moment of it. I guess to your second part of the question is what gets me excited about research? I think for me, it's the ability to ask questions that you're interested in, but also to find answers and to learn about things that no one else in the world knows the answer to. And I think it's quite a privilege to be able to do that, and it's a very rewarding experience. We have so many tools and technology here in Toronto, and I think being able to ask and answer and learn about diabetes at a level that no one has ever learned about before is just extremely fascinating.
Great, thanks, Emily. And I know you've been really active in Medicine by Design's trainee programs over the last several years. I'm wondering if you could comment on how maybe 1 or 2 of these programs have impacted your graduate student experience, perhaps touching on not only the commercialization and entrepreneurship opportunities, but also those around communications.
Sure. Yeah, so I was part of a variety of Medicine by Design programs. One was a 2-week course in the summer. It was a translational workshop where they brought together trainees and scientists from not just within North America, but from Europe and I think Australia as well. And together we were able to learn about translating products and our research into a therapy. So that was really exciting, and it's definitely skills and knowledge that I don't learn about in my classes when I'm doing a PhD. And the other program that was actually extremely useful, not just for science, but in life in general, was a communication workshop where we were able to learn how to more effectively communicate our research to the public. Clearly, it's something I'm still working on and need to work on, but it's just skills that I was unable to get from other sources. I find through Medicine by Design, it's not just these skills that have helped me as a scientist, but it's the unique thing about medicine by design is it brings scientists together from different research areas and from different fields. And although we come together, let's say for a workshop to learn a specific skill, when you all are in one place together, you make connections and you collaborate with people. And I think it's these collaborations and connections that have actually been extremely instrumental in my thesis. And my thesis project has taken specific turns from collaborations I've met and made at Medicine by Design events.
Thanks, Emily. We'll hear more about the really cool research that you're working on in a few minutes. Sean, what about you? What lab are you working in? And tell us a little bit about how far along you are in your degree and what gets you excited about research and innovation.
Yeah, so I'm in Professor Sefton's lab, which is working in regenerative medicine. But similarly, I kind of was pulled into that area of diabetes research. And it was kind of along the similar lines as what Emily was talking about. It kind of hit me like a brick wall a few years ago that, you know, there are a lot of ways we could improve on therapies for type 1 diabetes, and especially that kind of idea of regenerative medicine. And so I kind of pursued that in the lab because that was one of the avenues it was going in. And one of the cool things about that kind of ability to discover new things, I mean, it's a similar answer, but it's kind of cool to be able to put a lot of work in and actually discover something that not only people didn't necessarily know, but people might actually appreciate. So from that same kind of collaborative aspect, when you go and talk to people about the things that you spent a lot of time to do, they actually care and they might actually use them, which is really cool. And then it's got that extra little bonus and it really motivates you because this is in the context of medicine. So you might have that really positive impact on some people's lives. So it's just kind of a great overall motivator to do some research.
Thanks, Sean. And I know you're a chemical engineer, as am I. And often when I tell people that I'm a chemical engineer, but that I work in regenerative medicine, they look at me a bit funny and think, well, that's weird. How'd you end up there? How is your training, if you look back on your undergrad training as an engineer, how's that helped you or enabled you to approach your PhD from a unique perspective?
So I'm tempted to talk about unit operations, but I'll skip that. The chemical engineering perspective was kind of nice because instead of kind of getting focused on individual components that are really important and are breakthroughs, the chemical engineer kind of just goes and looks at what's available and they put it together, or they try to put it together. And that's the whole study, is how can I actually take all of those cool breakthroughs, combine them with the knowledge of what the problem is, and really put it together and try to implement it, which again really complements that kind of medicine by design strategy.
Wonderful, thanks. So Sarah, over to you. I know that you're again coming from a slightly different background from Emily and Sean as an immunologist. Tell us a little bit about where you are in your journey as a graduate student.
Yeah, so I'm in my second year, but I did complete a master's before starting my PhD in Dr. Sarah Crom's lab. And what kind of got me interested in research in general was the immune system and the potential for harnessing the powers of the immune system in translating those different capacities of the immune system into therapies. And in specific, why I'm so excited about my research is it focuses on harnessing capacities of the immune system that we don't generally know to think about the immune system. A lot of times people equate the immune system to inflammation, but there are many new discoveries that have been happening that show the immune system is also really important in homeostasis. And so that could be really important in implications for autoimmune disease and specifically, of course, in type 1 diabetes, but it has, broad implications in transplant as well as other autoimmune diseases as well. And kind of what gets me excited about every day going into the lab is just the potential for any of the knowledge that I gain or am able to discover contributing to any therapies down the line and potentially, being translated into medicine that the people that need it are able to benefit from.
Wonderful. Thanks, Emily and Sarah and Sean for that great introduction. So before we dive into each of your projects, I wanted to just pause and let all of the folks in the Zoom room know that if you have a question for our panelists, happy to have you enter your question into the chat, and we'll take questions at a couple of points throughout the hour. Where, you know, anything and everything is on the table in terms of questions. What I've asked Emily and Sean and Sarah to do before they talk about their research is to set the stage for regenerative medicine approaches in the context of current treatments for diabetes research. So Emily, I'll pass the floor over to you to start painting that picture for the audience.
Sure, thanks, Allison. So as I'm sure you're very much aware, when someone has type 1 diabetes, it means that they don't produce insulin. And the cells that produce insulin, just so we're all on the same page, are called beta cells. So if someone has type 1 diabetes, they don't have beta cells because they've been destroyed or attacked by their own body. So the main form of treatment, as you know, and which is why we're all here today, is because insulin was discovered, and most patients are actually using exogenous sources of insulin. So you can see here that people will inject insulin, as you're probably aware, but there's also been advancements to the technology that's available for patients. And such advancements include the insulin pump, as well as a device called a CGM, or continuous glucose monitor. These devices can actually be now used together in an artificial pancreas-like system and can deliver insulin automatically based on the patient's blood glucose levels. Although these forms of therapy, and although insulin is great and it's actually saving people around the world who live with type 1 diabetes, we know it's not a perfect therapy, and people who have type 1 diabetes still suffer from both hypo and hyperglycemic events. There is one other form of therapy you can see at the bottom corner is whole pancreas or islet transplant. So just so we're on the same page again, so the insulin is produced by a beta cell, and a beta cell is actually located within a group of cells called the islets. So the islets are just all of the hormone producing cells, and islets are located within the pancreas. So patients can either be transplanted with an entire pancreas, which contains beta cells, or islets, which also contains beta cells. And it's actually this form of therapy is able to completely restore someone who lives with type 1 diabetes, restore their blood glucose levels. But this therapy is not perfect. One, the patient needs to be on immunosuppression, which is not ideal, but also there are not enough donors over the world to cure everyone who has type 1 diabetes. And this is going to be a problem if we want to help these patients. So one approach to overcome this problem is to use a special type of cell called a pluripotent stem cell. A pluripotent stem cell has two characteristics. One, it can divide indefinitely, so therefore we should have an unlimited supply of cells. And this could overcome the lack of donor situation. And the second characteristic is that a pluripotent stem cell can actually become any cell type of the body. So what scientists around the world are trying to do is to take these pluripotent stem cells and to turn them into those insulin producing beta cells. This way we could have an unlimited supply of cells that could ultimately be transplanted into patients with type 1 diabetes.
All right, that was a great introduction into islet transplant. And I just want to acknowledge that while the project that I'm going to kind of introduce you to looks at islet transplant and potential regulatory immune therapies. There are a lot of other groups looking at regulating the immune system actually before this need is needed, but this may not be a viable option moving forward for everyone. And so in some cases, islet transplants may be needed. And right now, there are a couple of different options for islet transplant. So the more traditional method that Emily touched on was taking a pancreas from a deceased donor, isolating and purifying the beta cells and infusing these into the patient. The other option is, Allison, if you want to just to the next slide, is taking the pluripotent stem cells from the patient themselves and inducing these into stem cell derived beta cells, and again, infusing these into the patient. However, each of these transplant methods may be improved by the addition of a cellular therapy in the form of a regulatory immune cell. And specifically, the work that I focus on is introducing innate lymphoid cells, or ILCs, as a cellular therapy in combination with islet transplant. And this may have broad effects. For example, regulatory immune cells could have tissue regenerative properties, in such they may help islets engraft or repair. And in addition, it may decrease the immune invasion against islet antigens and suppress any graft versus host responses that may occur without requiring the need for immunosuppressive drugs after islet transplant.
Okay, so once we actually have all of those cells that we want to transplant, we have to look at that kind of step 3, the infuse into patient. This is actually a complicated thing, because unlike maybe something like a heart, you can't just kind of replace what's there. So you can't infuse those directly back into the pancreas, because they're normally kind of scattered all around. And so what people have started to do is use what's called the Edmonton protocol. So it was developed obviously in Edmonton, which is pretty cool. And what we do here is we would take those cells that we need to transplant, and you kind of clump them all together and then put them into the portal vein, which is a big blood vessel that goes through your liver. And they did this because it gives the cells all of the oxygen and the nutrients they need, and they're able to actually put insulin into the body. But it's a little bit limited because being in direct contact with the blood actually hurts it. And so that's one of the reasons we need so many donors and so many transplant cells, is a lot of those delivered cells die, excuse me. And so the regenerative approach that we're kind of trying to move towards, which is that kind of tissue engineering, we're just going to scoop up all of the good research other people have done and put it together, is something like an artificial organ transplant, but where we're actually using biological components. And so there are a bunch of different strategies for how to actually do this, but one of the common ones, and one of the ones that we do in the lab I work with, or I work in, is to take those beta cells and take those immune regulating cells or immune regulating kind of proteins, and combine them with these synthetic polymers, and we put them together and we create this new kind of artificial organ. And once we have that new organ, we can actually go and we can transplant it to places that might be a little bit better than directly into the portal vein. And that's actually one of the things we consider when we're designing them, is where do we actually want to put them? And so we have the options to put them in a bunch of different sites. And so one of them is in the peritoneal cavity, where a lot of your other internal organs are kept. But another one of the kind of interesting sites is just directly under your skin. And so those are the kinds of the things that we're trying to look at moving forward.
Great, thanks everyone for painting that picture of the current diabetes landscape, and where both cell-based and biomaterials-based strategies for diabetes are being deployed and developed. So I'd like to dig into a little bit more around what each of you is working on. I've got a few specific questions based on each of your research, and then we'll have a discussion around the impact that you hope to have based on the various product concepts that are being developed in your lab. So Emily, why don't we start with you again? You spoke a little bit about what you're working on, but maybe you can tell us a little bit more of about your research. And my specific question for you is, how do you know how to turn a pluripotent stem cell into an insulin producing beta cell? Sounds pretty tricky to me, so I'm sure the audience would be curious to know how that's done.
Sure, thanks, Allison. So I'm, again, in Dr. Nostro's lab, and so the goal of our lab is to take a pluripotent stem cell and to turn it into a beta cell, as Allison mentioned. And just to remind you, a pluripotent stem cell has two characteristics. One, it can divide indefinitely, and the second is that it can become any tissue or any cell type of the body. So the approach that we take to turn a pluripotent stem cell into a beta cell in the lab is to actually copy what happens during normal human beta cell development. And the reason we want to do this is because all of us, you and I, we actually started off as pluripotent stem cells at one point in time. When we were an embryo at the very early stage, the cells in an embryo are actually pluripotent, So you have an embryo, and 9 months later, you get all cells of the body, including beta cells, which are located within the pancreas. So we want to use this logic or this idea and kind of mimic or copy it in a dish in the lab. We know that as cells develop, or as humans develop, it takes 9 months. Things don't happen immediately. You don't go from an embryo into a pancreas the next day. Everything happens in stages. So what we try to do in the lab is we try to take our cells through these different stages that a cell would normally go through when a pancreas and when a beta cell develops. So the tricky part, however, is, so we want to copy what happens during human development, but we don't actually have access to human fetal samples for ethical reasons. So this is tricky because we need to try to figure out essentially what to feed our cells to bring it through these different stages. And the way that we do this is we actually learn a lot from other model organisms, such as mice or rats or zebrafish, and we can learn how beta cells develop in these organisms, and then try to see if that same concept can be applied to the stem cells and to the pluripotent cells we have growing in the lab. So right now, a lot of my work is, so I spend a lot of time reading to learn from other scientists around the world and to learn from other model organisms, and then try to apply this knowledge and try to test out a lot of different compounds in the dish in the lab.
Thanks, Emily. And if you think about your research, directions beyond when you finish your PhD, which as I understand is going to be hopefully within the next few months.
Yes.
5, 10, 15 years from now, as this research program in Christina's lab continues, what do you hope the impact of this research will have in terms of patients and how diabetes may be treated?
So right now there are actually clinical trials going on that are using pluripotent stem cells, and they're being transplanted into patients with type 1 diabetes. So already we kind of see that step forward. Right now, these clinical trials aren't going to cure diabetes yet because we still have a lot of work that we need to do. So I think I hope within the next 5 to 10 years that we can have a reliable cell product that was generated from pluripotent stem cells that could ultimately be used for therapeutic purposes.
Great, thanks, Emily. And that's probably a great segue into Sarah's work, because Sarah, I know that you're working very much on the immunoregulatory components of a cell-based therapy. So my question for you is, what is an ILC? And why are these cells important in developing a cell-based therapy for diabetes?
Yeah, so an ILC is the short form for innate lymphoid cell. So these cells are pretty newly discovered, and they're immune cells, which are primarily tissue resident, and they are known now to have broad functions in the body, although we're still characterizing, exactly how they work and how they affect each individual body and tissue organ. And so there are still several subtypes of ILCs, including natural killer cells, ILC1s, ILC2s, and ILC3s, and each are kind of characterized by the molecules in which they secrete and, the functions that they perform. And so some of the functions of these ILCs, researchers have found to be immunosuppressive as well as tissue reparative. So their regulatory functions may be important in type 1 diabetes, as type 1 diabetes is an autoimmune disorder in which the immune system actually attacks the beta cells, and that results in beta cell death. This is propagated primarily by T cells. And if we can understand and figure out a way for ILCs to suppress the T cell function, then perhaps they can change the microenvironment around the islets or the beta cells, allowing them to survive in the body. And secondly, they also have tissue reparative functions. And so this may be important specifically in combination with islet transplant because it may help for the islets or the beta cells to engraft better once they are infused into the body. However, the role of ILCs right now is largely unknown in type 1 diabetes and specifically in the pancreas. So right now, we're looking to understand the role of ILCs specifically within the healthy pancreas. And then, you know, if they do have kind of tissue reparative and suppressive functions within the pancreas, then we may be able to use and harness these functions to support islet transplant and potentially suppress any negative effects by type 1 diabetes or graft versus host disease.
Great, thanks Sarah for that overview, sounds fascinating. What do you think? I know you're just in your second year of your PhD, so you have a few more years to go, but if you sort of cast out a 10-year lens on your research trajectory, what do you feel the main or one of the main impacts of your program might be on the field?
Yeah, well, we generally right now, there are a couple of ways in which my research may contribute to the field. And the first is just understanding if ILCs are a potential viable cellular therapy, regardless of whether they are kind of in conjunction with islet transplant or not. And then also characterizing better the role of ILCs within type 1 diabetes if there is dysregulation which occurs during type 1 diabetes in the actual populations of the ILCs themselves. And then, you know, hopefully taking whatever knowledge is gained and translating that into potential cellular therapies.
Wonderful, thanks Sarah. So Sean, shifting gears a little bit to talk about the tissue engineering approach, how are you combining the use of synthetic biomaterials in cells to create this artificial pancreas approach that you talked about in your introduction?
Yeah, so I'll just do the quick recap. My project's kind of looking at how those materials can be used to combine the cells, and when we're studying them, we kind of have two main metrics or things that we care about. And that's whether the insulin producing cells actually, lower blood glucose levels. And so we monitor how well they engraft. And then also monitoring the immune response to the cells and trying to incorporate some things to protect them from the immune response. So it's kind of a nice combination of everything. Like I said, the chemical engineers like to do. But the kind of cool thing about the synthetic polymers we're using is they serve a few purposes. The original kind of stepping back idea was that those polymers would be what you actually use, and we called it a scaffold, like the bare bones of that artificial organ you're going to develop, and then you would use that to kind of arrange the cells as you needed, and then use that to transplant them into the cell into the body. So that's kind of the main purpose. But the cooler thing that we've been finding more recently, and why we kind of call them more regenerative biomaterials, is we found that they actually can talk to the host, and if you use the right kind of combinations of molecules, you can get it to ask the host basically to help engraft that new artificial tissue, which kind of speaks to one of the themes from Medicine by Design a bit earlier, the idea that we could try to work with the host as opposed to trying to kind of fight off the immune system. And so it's a cool material to include because it really tells the host, oh, maybe I want to actually keep this here and hook it up to my internal systems.
Thanks, Sean. And what about you when you think about sort of the trajectory of this research? I know the biomaterials approach in the Sefton lab has been ongoing for a number of years, but where do you think your work will lead in the next 10 years if you think about product concepts and potential therapeutic approaches? Yeah,
so maybe two kind of avenues. One of them would be improving that kind of maybe how clinically practical it is to do these islet transplantations. Because if the study goes well, it'll be highlighting that maybe we can improve the efficacy or reduce the number of cells that die when they're transplanted, which would make it maybe a more approachable therapy or a more common one. But then the second kind of thought there is those interesting synthetic polymers that have those regenerative properties. They might kind of change the way tissue engineers are looking at things and switch it to that more mindset of, okay, how do we actually work with the responses that the host can give, as opposed to trying to just hide it and keep it alone and hidden from the host.
Thanks, Sean. So great overviews of your research, and certainly, you know, some very synergistic approaches to developing strategies to treat diabetes. I'm just going to pause for a moment to ask our audience if they have any questions for Emily, Sean, or Sarah. If you do, you can enter them into the chat function in Zoom, and we'd be happy to answer any questions. So maybe I'll just pull one off that just came through. And this one is a question for Sarah. Sorry if I missed it, but do we have any hints of whether ILCs play any role in other autoimmune diseases or transplantation?
Yeah, for sure. So I guess I'll back up. So I'll start first on autoimmune diseases. It's a fairly new field, and whether they play a specific role in propagating autoimmune diseases is not necessarily clear. However, there have been many studies in mice specifically showing that if you increase the amount of a specific type of ILC, ILC2, that you can reduce the symptoms of arthritis and even, sorry, the other autoimmune disease is escaping me, but there have been studies that have shown that basically if you induce the expansion of some of these subtypes of ILCs, there can be a better outcome. Sorry, no, still don't remember it. And then specifically in transplantation, it's kind of the same thing. So there have been also studies of if you give specific injury to the kidney, and then you also put in ILCs at the same time, a specific type of ILC, ILC2, that you can ameliorate some of the downstream repercussions that's associated with the injury. So not so much in humans yet, although there was one study that looked at the amount of ILCs in type 1 diabetes, and a more kind of suppressive alteration was shifted in patients with type 1 diabetes to have a more inflammatory ILC immune profile, and then in healthy, they might have a more suppressive immune profile. So yeah, there's definitely a lot of literature out there, and we're just looking now more into the human side of things.
Thanks, Sarah. Any other questions from the audience at this point? Okay, well, maybe what we'll do is we'll move on to the next session of the panel discussion, and we can pop in with questions as they come in. So shifting gears a little bit, you know, we've talked about why regenerative medicine is exciting and the work that you're doing in your labs. Let's talk about the future. I know it's always a sort of a scary thing to think about and to talk about, but I'd be really interested in understanding from each of you, what are your goals and your future aspirations, not only for your research, but what comes next after your graduate degree, and where do you hope to be in 10 years? I know those are loaded questions, but always interesting ones to discuss. So why don't we mix up the order this time? And Sean, we'll start with you.
It's the hardest questions. So where would I want my research to be in the future? So it would be nice research-wise if these kinds of therapies or those improved transplant protocols are actually implemented. And so that would be where I hope the research goes, and also where I hope to be working is going and developing and improving those protocols, and then making them actually clinically feasible and working on how to implement them as a more widespread therapy. So that would be kind of the research end. The personal end, I don't know. I've got a few years left on the degree, but after that, maybe it depends really where the field is at. So regenerative medicine is really cool, obviously. And so if there are things like industrial opportunities to actually go and pursue those regenerative therapies in a way that would be meaningful towards that clinical translation, then that might be where I go. But If not, then staying in kind of an academic setting and trying to research them and really make it so that it's something that we could approach clinically.
Thanks, Sean. And certainly listening to the panel that was before us this afternoon, you know, we heard the sort of the history of how Toronto and the University of Toronto has continued to grow to support commercialization and translation. And we have companies now like Blue Rock Therapeutics and AvroBio and others that are, you know, have wet lab facilities here just adjacent to the university. So I think as a graduate student coming out with a degree in regenerative medicine, there's no limit to the opportunities, both within and outside of academia. So great to hear that your eyes are open to those opportunities. Sarah, what about you? When you think about future sort of aspirations, I can imagine, I think back to my PhD days, and certainly when you're in your second year, that's sort of your core focus, is that runway between now and defending your thesis. But interested to hear your thoughts on the future of both your work and where you think you might head.
Yeah, so certainly early on in my PhD, but just in terms of my research, I'd like to see, you know, really determine whether ILCs could be viable to support islet transplant. There have been some studies specifically looking at mice that have shown that ILCs would be viable, but we want to show it with human cells. So hopefully accomplishing that goal, and then, you know, in general, just learning more about how, you know, there are dysfunctions within type 1 diabetes and what we can do to kind of like bring that back to more homeostatic levels. Looking forward to my future career progression, I'm not too sure what is going to come after my PhD, but I mean, right now I'm thinking about a postdoc and then potentially going into academia or biotech, as Sean had mentioned, and, you know, working in this realm of regenerative medicine. And I'm really interested in how we can harness the capacity of the immune system, because it's so, I mean, it's amazing. It helps us live every day and, you know, overcome so many different obstacles. And so how we can harness that for therapies, I would love to work on for, you know, a very long time.
Sounds like a fascinating endeavor for, like, keep you busy for decades to come. Emily, what about yourself? You're sort of at the closest edge of your PhD of our panelists. What are your thoughts on the future?
So I guess scientifically, yes, of course, I'd like to finish my thesis project. So I have two projects. One is my main thesis project, and the other is developing a protocol to make or improve beta cell differentiation. So I really hope that not just my research, but in the field, that beta cell differentiation protocols are improved quite dramatically, so that we can have a therapy. And as it was evident from this panel, that for a therapy, we don't just need cell product, we need to properly regulate the immune system and properly have ways to transplant these therapies. So I hope in 5 or 10 years that we can combine all of these, or all of the knowledge that we will acquire over this time, to have a therapy that kind of answers all of these different questions that we're looking at. In terms of my future, so I'm graduating soon, so I should have a more definite answer, but I don't. I really enjoy diabetes research, and so I hope to stay within this field and be involved in the processes that are involved in moving research into the clinic.
Thanks so much, Emily. We have a great question from the audience that I think each of you can offer a perspective on. The pandemic has been really hard on our researchers, especially the trainees, as it limits travel, opportunities to meet others, sparking those spontaneous collaborations. How are you overcoming this through the pandemic? And what more would you like to see happening as opportunities to support this? Happy to have anyone jump in.
You know, for me, although we have some collaborators in other parts of Canada, But one of the really nice things about being in Toronto, I think specifically, is you have just such a wealth of diverse backgrounds right next door. So for like specifically, I've been able to collaborate with the Nostro Lab, and they've been able to show me lots of techniques working on islet biology and how I can incorporate those into my research project. And so in that way, the pandemic has been hard, but it hasn't, been unbearable. There's still the supports needed for sure to get my research done. And I guess this to the second part of the question, what opportunities? I don't have any, specific things because, the pandemic is so, who has the answers for those? But I think like so far, the University of Toronto and, they've been doing a great job to support trainees throughout the pandemic. So no complaints.
Emily, on anything from you?
Sure. I found actually through the pandemic, I've been able to connect, or people have been quite open to connect and to meet with me virtually. So right now I'm obviously trying to figure out exactly what I want to do next. So I've been able to actually still meet with PI, scientists, people in industry through virtual meetings. So that's been actually quite helpful for me. And I think in order to maybe provide more opportunities, if students, specifically trainees, are maybe more open or aware of potential people who would be willing to meet with us. Because we have conferences still, but it's really hard to, let's say, connect with a PI or meet someone at a virtual conference because there are thousands of people there and you're not going to walk by them, you're on a computer screen. So maybe just making those opportunities maybe more available would be quite helpful.
That's a really great suggestion, Emily. And as you know, the Medicine by Design annual symposium is coming up in a couple of weeks. And, we're looking at having upward of, 500 to 700 people from around the world participating. And one of the things that we're struggling with right now is how do we establish meaningful virtual, bump-ins that you would have outside of the Mars auditorium at an in-person conference. And certainly it's a unique challenge that I don't think any of us are used to navigating yet, but we learn as we go. Sean, what about you?
So I unfortunately don't have any better suggestions than what have come up already. I mean, I completely agree. That really encompasses it. But the fun little note maybe I'll add on the side is that, the shutdown kind of stopped our ability to do research for a while, but at least among the group I'm in, after a certain point, we just were sitting around thinking, and that's almost torture, but we started talking amongst each other and we really were kind of collaborating more, just bouncing ideas off each other. So, we weren't actually stopped too much. It at least let us think about what we wanted to do and try to hash out those experiments. So, it hasn't been as bad as it could have, for sure.
That's great to hear. I have a final question for everyone, and I'll keep my eye on the questions from our audience members as well. I often like to ask this question when I'm interviewing our PIs, but I'll ask a modified version to you. If you were to look back 5 years and give yourself advice, what would that advice be? And Emily, maybe we'll start with you.
Sure. I guess, I had to go through the process to learn how critical this was, but I think it's nice to know now, I would have liked to learn earlier, that it's okay to stop projects when they're not working out. Part of science isn't to get all of the results that you expected and to get good results all the time. I got a lot of bad results. I've had to jump projects quite a few times because things weren't working out. And if I had the knowledge 5 years ago to know that it's okay and normal that projects don't necessarily work out, I may have saved a year early on by jumping and switching projects then.
Thanks, Emily. Sarah, what about you?
Yeah, so 5 years ago, I was I think, a third year in undergrad, and I was actually just starting to look for opportunities to go into immunology labs and get my hands on, the first little bit of research that I ever did. And the thing that I remember the most about me 5 years ago in a research environment is that I was so nervous. And I did not have any confidence that I could go into a room of such like smart, capable people and kind of be on that playing field. And so I think what I would just tell myself 5 years ago is that have confidence in your abilities, because you're capable of, being a grad student and getting research done and being successful. So, yeah.
That's a very wise piece of advice. Sean, what about yourself? What would 5 years ago Sean have said to you?
I don't know. That's the hardest question. Again, they just keep getting harder. Again, I was also in my undergrad. I think in third or fourth year chem eng at Waterloo. So I might have said something like, relish your life when the most difficult system you have is this little distillation column, because it's nothing like the biological complexity of the work we do now. But from an actual better advice perspective, it might be to go and kind of connect better with research groups or professors kind of earlier and earlier on, because even if it's not your specific area that you wind up going into, those connections are pretty valuable, and it would have been nice to make probably a few more back then.
Great, all wise words to your former self. I wanted to take the opportunity as we close out this panel today to thank each of you for participating, sharing your research, sharing your vision for your projects and the impact that your collective research efforts will have on the field of diabetes treatment. Certainly, as a member of the Medicine by Design leadership team, I mean, trainees are the lifeblood of our program. Without trainees, we would not be Medicine by Design. So I hope that all three of you continue to engage with the community, continue to reach out to myself and the team with ideas and with opportunities, and that we can support you as you move your projects along and as you get more and more engaged in the translational aspects of your research. With that, I'll perhaps pause and just see if there are any final questions from the audience at this time. Looks like all is quiet on the chat function. Emily, Sarah, and Sean, any questions for me?
Allison, what would you tell yourself when you were starting your PhD?
I had a feeling someone would turn this around on me. I think, Sarah, your advice actually really resonated with me when I look back. So I also was a chemical engineer undergrad and started noodling around in research as a summer student just after third year. And I also had, just didn't have the confidence, sort of assumed that everyone was smarter than me and had some sort of edge that I hadn't figured out yet. So I think too, if I could have gone back and tell myself, don't be so worried about making mistakes. And also, it sounds a bit sort of cliche, but seize every opportunity you can to network. I know back when I was a student, and I know in speaking with folks like yourself, it's a daunting prospect, particularly if you're thinking of working outside of an academic career path. But there are just so many opportunities out there. I mean, Toronto biotech is booming. And so I would just have told myself, even back then, to network. You never know when you're going to meet someone that has a job, that's looking for someone with your skill set. And serendipity can be a really powerful thing. So I think that would be the piece of advice that I'd give myself. And that's maybe something I should continue to remind myself moving forward in my career.
Allison, where do you see Medicine by Design
that
in
was
10
mine.
years?
So where do I see Medicine by Design 10 years from now? Is that what you were? So another great question, and as many folks may know in the audience, Medicine by Design's funding started back in 2015. And so our funding from the federal government will come to a close in a couple of years. So I'm working with our leader, Michael Sefton, and the university on a strategy for what we're calling Medicine by Design 2.0. And I think, you know, if I think about Medicine by Design 2.0 and where I'd like us to be in 10 years, I think continuing to broaden the scope of regenerative medicine. Cell and gene therapy has so much potential, but I think that other complementary areas such as endogenous repair and using small molecules to harness the body's ability to regenerate itself is a huge opportunity. I also think that we can continue to use models, stem cell models, to understand disease. But really what my hope is that 10 years from now, we can look back and say that we've launched another couple of Blue Rock therapeutics. You know, we had this huge investment in Gordon and Mike Laflamme's work several years ago. And I know for Medicine by Design, we want to continue to invest in research, but also in translating that research and helping grow the biotech ecosystem in Toronto so that trainees like yourself can graduate and actually have jobs in industry if they so desire. And those jobs can be not only jobs in the lab, but also leadership jobs as you move through your career. So that would be my hope for Medicine by Design. And certainly we're planning on being around for more than 10 years. Well, I think that's probably a great place to end. And thanks to our panelists for putting me on the spot as well. Thank you all. And thanks to the audience for listening in. I hope that this gave you a meaningful window into the work that Medicine by Design is doing in the diabetes space. Have a good evening, everyone.
Thank you.
Thanks.